Buenos días a todos y bienvenidos. Vamos a dar comienzo a la conferencia del doctor García Sastre. Even if he's Spanish, I'm going to do the presentation in English because his presentation is going to be in English as well. And just in case there are people that do not understand Spanish. First of all, I would like to thank Adolfo for waking up so early because he's in New York and it's around 8, and we've been chatting for a few minutes before the start of this talk. Professor García Sastre is professor in the departments of microbiology and medicine and director of the Global Health and Emerging Pathogens Institute of Icahn School of Medicine at Mount Sinai in New York. For the past 30 years, his research interests have been focused on the molecular biology of influenza viruses and several other negative strand RNA viruses. He started with his postdoctoral training in the early 90s. Well, I forgot to mention before that he was born in Burgos, and he got his degree in biology and his PhD at the University of Salamanca. And then he went for a postdoc to the Mount Sinai with a postdoctoral fellow on fellowship from the Icahn and then from the Fulbright program. And he started his training. During this training, he developed for the first time novel strategies for expression of foreign antigens by a negative strand RNA virus, the influenza virus. He has made major contributions to the influenza virus field, including the development of reverse genetics techniques allowing the generation of recombinant influenza viruses from plasmid DNA, in collaboration with Dr. Calese, the generation and evaluation of negative strand RNA viruses vectors as potential vaccine candidates against different infectious diseases, including malaria and AIDS, and the identification of the biological role of the non-structural protein NS1 of influenza virus during infection. His studies have provided the first description and molecular analysis of a viral encoded EFN antagonist among the negative strand RNA viruses. And these studies have led to the generation of attenuated influenza virus containing defined mutations in their EFN antagonist in protein that might prove to be optimal live vaccines against influenza. His research has resulted in more than 500 scientific publications and reviews. He's the director of the Center for Research of Influenza Pathogenesis, one of the 5 NIAID-funded Centers of Excellence for Influenza Research and Surveillance. He was among the first members of the Vaccine Study section and member of the Virology B Study section of the NIH. In addition, he has served for 5 years as editor for the Journal of Experimental Medicine and is editor of PLAS Pathogens, Journal of Biology and Virus Research, and member of the editorial board of Biology, Vaccine, NPG Vaccines, Current Topics in Microbiology and Immunology, and Influenza and Other Respiratory Diseases. He's a member of the Scientific Advisory Board of Keynote Symposia. He has co-organized, or he is the co-organizer of the International Course on Viral Vectors held in Heidelberg, Germany, and the first research conference on orthomyxoviruses held in Icahn, the Netherlands. Also co-organizer of the 7th International Society for Vaccines meeting in 2013 and the Keystone meetings in 2014 on respiratory virus pathogenesis, and in 2017 on interference. His publication in Science, in the journal Science, on the reconstruction and characterization of the pandemic influenza virus in 1918 has been awarded with the distinction of the paper of the year in 2005 by The Lancet. In 2000, 2005, he became a fellow of the American Academy of Microbiology, and in 2009, he received the Beijing Professorship from the National Academy of Sciences of the Netherlands. In 2011, he has been elected president of the International Society for Vaccines for the period 2014-2015. In 2017, he was elected a fellow of the Royal Academy of Pharmacy in Spain. In 2019, he was recognized with a honorary doctorate degree from the University of Burgos. And in 2019, he was elected a member of the National Academy of Science of the United States. So thank you very much, Adolfo, for giving us this talk. We are all excited to hear about the new COVID-19 therapies and vaccines. I'm going to put, I know there are many students connected that would like to receive a certificate of assistance. So we have put this QR, I don't remember the name, but so you can scan it and you will receive your certificate. We will put it also at the end of the talk. So now I will stop sharing my presentation. I will leave the word to Adolfo. During the talk, you can place your questions in the chat, and we will ask at the end of the talk. Okay.
Okay. Thank you very much. Thank you for being here and listen to what I'm going to share with you, which is mainly research that I've been doing in my lab in collaboration with multiple labs in the areas of therapies and vaccines against COVID-19. And let me share my screen. Okay, so I'm going to start with my presentation, and what you see here is an early picture that we got in virus-infected cells. In green, you see ACE2 expression, which is the receptor for the SARS-CoV-2, and in red, you see one of the proteins of SARS-CoV, and these are cells that are susceptible for infection with SARS-CoV-2 that help us to develop assays for looking for antivirals early on when the virus started to circulate in humans. And these allow us to advance quickly as well. The other thing that helped us to advance quickly was a large collaboration between multiple groups in this area. Okay. So I'm going to talk first about therapies and what strategies there are for therapy. There is already an approved therapy for the treatment of COVID-19 patients. That's remdesivir. Remdesivir seems to work mainly when people can get remdesivir, which is an inhibitor of the RNA-dependent RNA polymerase of the virus, when people receive the treatment early during infection. After hospitalization and severe symptoms, it seems to be a little bit late for remdesivir to be able to make an impact, and that's why WHO has said now that remdesivir perhaps is not the adequate treatment for people that are already hospitalized. But still, it's a good treatment if you can treat people early on during infection before they develop severe symptoms, because it stops virus replication. Now, it has the disadvantage also that it's a very expensive treatment, as well as it needs to be given IV, and therefore not everybody can afford the possibility of getting remdesivir. There are some other treatments that are approved for emergency use, like the use of plasma from convalescent individuals with antibodies that will inhibit also virus replication. The use of monoclonal antibodies, the Eli Lilly monoclonal antibody has been approved for use in emergency use in the United States. These are antivirals like remdesivir, and like remdesivir, they are much better working if they are given early during infection. And then dexamethasone, which is an immunosuppressor, has been also approved for emergency use for people with severe symptoms. It seems to give an improvement in a percentage of people with severe disease. And in this case, when one is treating is the symptoms, not so much virus replication, and that's probably why you need to some type of immunosuppressant at the end of infection when there is not so much virus replication, but there is a lot of immunopathology, and that's what you want to stop. However, I think that we can do better than that, and we need still to find better antivirals that can be given to people. Even if the vaccines are coming, most likely the virus will still circulate, not causing so many problems like what is causing now, but still some people will have severe disease, and it will be good also to have good treatments for people that get severe disease, even if the vaccine is working well. And then there is a huge reduction in this number of cases, because still there will be some cases that will be hospitalized, and they may get severe disease once that the vaccines are starting to be used. So my lab and many others have used a strategy for looking to new drugs, what is called drug repurposing. The idea in drug repurposing is, instead of doing huge screens for new drugs, one looks into compounds that they are already used for some indication in humans. They have been already through clinical trials, or they are used for the treatment of some disorders in humans. The idea behind is that these compounds will hit specific host factors that are involved in these specific syndromes for which people are being treated. It could be cancer, it could be allergies, it could be mental diseases, and that these factors may actually participate in, some of these factors may actually participate in virus replication, because viruses need to use host factors in order to be able to replicate. So the approach that we have been using is not so much, and we collaborate also with other people that have done screens of whole libraries of drugs that have been approved in humans. But we have been focused most of our efforts to predictions among the drugs that are being used in humans, based on the molecular biology studies, especially systems biology studies. And then coming from these predictions, then we take compounds, a limited number of compounds. Right now we have tested around 800 compounds. And then we look which ones are able to inhibit the virus in tissue culture. And then from that, we are moving now to animal models, and hopefully also to clinical studies that can be implemented quickly, because they are drugs that they are known in terms of toxicity and PK properties in humans, because they have been used for another indication. So there have been already clinical trials that have been done with these drugs. Okay, so the question here is, can we identify compounds that inhibit host factors involved in SARS-CoV-2 replication among non-human drugs? And the first thing that we did is, what host factors are likely to be implicated in SARS-CoV-2 replication? And for that, we collaborate with Nevan Krogan at the UCSF. It's here in this picture, that early on, when the virus started to cause problems in China, and when the sequence was released for the virus, then he cloned each of the individual proteins that are encoded by the virus, around 29 different proteins, expressed them individually in human cells, and then immunoprecipitate them, and then look to what host factors come with these proteins. And then he came with a map, interactome map, what is called of protein-protein interactions between viral proteins and human proteins. Basically what you have here in this little cartoon is each red dot represents a viral protein. It's too small to see which viral protein is, but the concept that is here is that it interacts with all of this constellation, each one of host proteins that are in total of 332. And because these are host proteins that interact with viral proteins, the assumption is that many of them are the reason why the viral protein is interacting with is because it wants to use its function in its own benefit, yes, to promote replication. So we think that this will be enriched for host factors that are required for viral replication. And then if some of these host factors, they have a known drug that is being used in humans for another indication, that has been in humans in clinical trials, then we test one of these drugs that are known to interact and antagonize some of these host factors for inhibition of our replication. And with that, we came out of these 332 protein-protein interactions with 69 what is called druggable host factors, that they have some specific compound that has been used in humans already that is able to inhibit the function of this particular host factor. And with that come the expertise of Kevin and Brian. These are chemical bioinformaticians that they look to these host factors and they identify drugs that are predicted to interact with these host factors, which is what you see here in this big also compendium of drugs. Again, in this interactome, there are now some drugs here put in here that are known to interact with and antagonize these host factors that are interacting with viral proteins. So they identified a total of 97 drugs, and we tested 97 drugs. That was mainly driven in my group by Chris White, with the help of Lisa, Elena, and Romel Rosales. So how they tested these drugs? So we put together an assay that is medium throughput to look for inhibition of replication of drugs in tissue culture of SARS coronavirus, in which we take 96 well plates. We put Viro6 cells, which are cells that support well replication of SARS coronavirus 2. And then after seeding the cells, we put the drug in these wells at different concentrations, the different drugs, 2 hours before we infect them with SARS coronavirus 2 in BSL-3 conditions. And we collaborate also with a group in Paris, in the Institut Pasteur, Marco Vignusi, Oliver Swart, and Christophe Danfard, that they are also doing these assays independent from us, and in order to see whether we get similar data. So we infect them in the presence and the absence of different concentration of these compounds. And then 48 hours later, the virus has replicated. It's an MOI that allows around for 3 cycles of replication. The virus has replicated in the absence of the drug. Then most of the cells are infected, and we can determine that with a antibody that is stained for the nucleoprotein. And if the drug is inhibiting viral replication, there will be less number of virus-infected cells found by the staining with an antibody against one of the viral proteins. This is the assay that we use, and then we confirm that this is actually inhibiting viral replication when we see a reduction in virus-infected cells by looking to the infectious material in the supernatant and see also is a reduction. And in addition, we have duplicate plates where we look for the cytotoxicity of the drugs. So we want compounds that are not cytotoxic and that are able to inhibit viral replication. And in Paris, they do a very similar assay. They use a slightly different MOI, and they mainly look to viral replication, not by number of infected cells, but looking to RNA viral loads by RT-PCR, and also plaque assay later on. And the good thing is that we got very good similar data between the data that we got in Mount Sinai in my group and the data that it was generated in the Pasteur Institute. So this, for example, a plaque assay to look for infectivity of SARS coronavirus 2 that was set up early on during the pandemic, as soon as we got the virus, by Lisa, Claire, and Randy in my group. And you can see that it's very easy to look for a viral titer because it makes this cytopathic effect that results in these clear plaques. And when you dilute it, then you know how many viruses you have in the supernatant. Okay, so how these assays look like? So this is, for example, this assay for antiviral activity with remdesivir, which we know is a known drug that inhibits the virus by inhibiting the RNA-dependent RNA polymerase. So what we obtain in these curves is we have in here in the discontinuous line is the cell viability in the presence of different concentrations of the drug and absence of viral infections. So we can see in this discontinuous line is that remdesivir is not killing cells, so it's not cytotoxic. And then even at concentrations of 10 micromolar, there is 100% cell surviving. Now, if we put now the virus and then we look for percentage of infected cells in the presence of different concentrations of remdesivir, we found that at high concentrations of remdesivir, there is no virus-infected cells. according to staining. And then as you reduce the concentration of remdesivir, then you start to get some replication, and then remdesivir stop working at concentrations that are below micromolar. So with that, we can get an IC90, which is an infectious an inhibitory concentration of 90% of our replication, and that's for remdesivir in viral cells is 1.5 macromolar, and also selective index, which is the ratio between cytotoxicity and inhibition. And you want to have this selective index as high as possible in the case of remdesivir, because it's not cytotoxic. We know it's more than 26, which is good. So that's how it works with remdesivir. If remdesivir, this is not very good in terms of inhibitors. You would like to have a little bit more sub-micromolar, but remdesivir actually is a very good inhibitor in human cells. So in viral cells, due to the presence of enzymes that need to make remdesivir into an active compound, that they are less abundant in these viral cells, it has an IC90 of 1 micromolar, but in human cells, and this is 293Ts, in this case, 293Ts are not infectable by SARS-CoV-2, but if we put the receptor AC2, they become infectable. Remdesivir has an IC90 very good of is sub-micromolar, around 20 nanomolar. And now these are some examples of the antiviral assays that we have done in my group. Multiple drugs, multiple compounds. Some are working, some are not working. And in total, we have now looked at around 800 tags and compounds screened, and find around 10% that are able to inhibit viral replication. So it's a good hit ratio. That's again because it's based on a rational approach to look for these compounds. And some of them are actually working at even better concentrations than remdesivir. So our first study resulted in 10 good agents that give us very good antiviral activity. And they could be divided into two categories. One was protein translation inhibitors like sotatafine or Ternatin-4, as well as Pitodepsin, which is a similar compound to Ternatin-4. We'll talk a little bit more about this one. And then the sigma receptor modulators. So this came from host factors, again, that interact with viral proteins. The sigma receptors interact with several viral proteins. These are chaperones that are not very well known what is their role in the biology of the virus or even of the cell, but there are multiple compounds that have been used for many different indications that they actually are able to inhibit these sigma receptors. Among them, for example, hydroxychloroquine, which is one of the drugs that in the beginning was thought to be used against SARS-CoV-2. And there are also antihistamines, antipsychotics, anti-anxieties, and some compounds that are in preclinical use. Among them, this compound PB28 is actually the one that gives us better activity among all of these compounds. So sigma receptors, they were identified to interact with ORF9C, one of the proteins of SARS-CoV, and with NSP6, another of the proteins of SARS-CoV. And these are different compounds: cloperastine, hydroxychloroquine, PB28, clemastine, haloperidol, or this PD144418. That, as you can see in the red curve, they are able to inhibit viral replication. And some of them are with little cytotoxicity, like cloperastine, and even the ones that are cytotoxic at high concentrations, they start to inhibit viral replication at a lower concentration. And as I mentioned, there are many different categories of drugs, like antihistamines, antipsychotics, antimalarials, even a hormone like progesterone, anti-anxieties, and some in preclinical development. So PB28 we think is a good compound for two reasons. One is this hydroxychloroquine is one of the drugs that are given us also hit here. So hydroxychloroquine is able to inhibit viral replication in vitro. However, it has also some problems with potential cytotoxicity because it not only targets the sigma receptors, but it targets a receptor that is present in the heart cells, this hERK, which is required for good function of the heart. and therefore there has some side effects that could cause cardiovascular problems. Now, when we look, our collaborators at USSF, when they look to the ability of PB28 to bind to sigma receptors here versus H-ERG, hydroxychloroquine and chloroquine, they bind well to H-ERG as well as to sigma-1 and sigma-2. But PB28 doesn't bind well to human ERG and binds well to sigma-1 and sigma-2. So it seems to have better specificity. And not only has better specificity, it has also a better IC90 inhibitory concentration, 90, that hydroxychloroquine. Hydroxychloroquine is bigger than micromolar, which you always want drugs that are lower than micromolar. And PB28 has an IC90 of 0.2 micromolar, 20 times better inhibitory activity than hydroxychloroquine. So we think that that could be a compound, and we are looking now to analogs of this compound to see whether we can improve their antiviral activity. The other category of compounds are translation inhibitors. This is one example, that's sotatyphine. This is a compound that is in phase 1-2 clinical trials for the treatment of cancer. It targets one of the translational components of the host machinery, the protein called EIF4A, that was found to also identify to interact with one of the viral proteins. And it has an IC90 of 144 nanomolar, so sub-micromolar. It has what it looks like some cytotoxicity, but it never goes to 0%. So it does not kill the cells. What actually happens is that because it inhibits translation in these cells, then it's cytostatic, meaning that the cells stop dividing, and then it looks like you have less cells 48 hours later than when you started, because in these assays, the cells in the absence of the compound, they double, and in the presence of the compound, they don't double. And therefore, when you measure the final number of cells in the presence and the absence of the compounds, you have half amount of cells in the presence of the compound that in the absence. So you score it as 50%, but this doesn't mean that it kill the cells. It means that it stops division. So it may not be as toxic as in vivo, and in fact is being used already in humans for potential use in cancer. And the one that we are quite excited actually is Pitodepsin, which targets a different host translation factor, which is EEIF1A. This is also involved in translation. And the interesting thing about this compound is that is extremely potent. So it has an IC90, both in viral cells and in human cells, around nanomolar concentrations, which is much better, for example, than remdesivir. And this compound was developed by a Spanish company, PharmaMar, that has been licensed for the treatment of multiple myeloma in Australia. So it's been already used in humans, and actually you require less amount for inhibiting SARS-CoV-2 replication than the amount that is required for the treatment of people with multiple myeloma. So the compound is also called Aplidin, and the target is EYF1A, and has an IC90 that is sub-nanomolar in human cells with a high selectivity index. So we think that that could be an important compound that may give an impact in virus replication. In fact, PharmaMar has already finished a phase 1-2 clinical trial for SARS-CoV-2 in Spain, and they are almost ready to publish their results. And I'm talking to them, and they seem to have very promising results in phase 1-2, and they are now trying to go for a phase 3 clinical trial. Okay, so that's with these protein-protein interactions, but the group of Nevan Krogan made some other predictions coming also with for what happens in virus-infected cells. And this came now with a second study in which he looked to changes in phosphorylation in the proteins that are infected in cells that are infected with SARS-CoV-2. And he looked globally, how many changes are in phosphorylation, and then based on these changes in phosphorylation, what are the predicted kinases that are being activated during viral infection that change phosphorylation. Again, the idea is the same with the protein-protein interaction. There is a kinase that is activated during viral infection. there is a good possibility that this kinase is activated because the virus wants this kinase to be activated in order to promote viral replication. And because there are a lot of inhibitors that are described of kinases, then we look among the inhibitors of kinases, that kinases that are found to be activated during viral infection, where they also inhibit replication of the virus. And that's what one sees in this slide. So these are some of the kinases that are predicted to be activated based on the phosphoproteome in virus-infected cells. And then one of them is this kinase, PIKFYVE, where there is a compound apilimod that is known to inhibit it, this kinase, and this compound also inhibit viral replication. There is CDKs that are being activated during viral infection. There are compounds that are known to inhibit the CDKs. And again, these compounds inhibit viral replication. There are MAP kinases that are being activated, and there are multiple inhibitors for MAP kinases. And again, these inhibitors are able to inhibit viral replication, both in our group as well as in the group of Paris. And in the group of Paris, also in this Pasteur, they did silencing from some of these kinases. And when you silence the kinase, you also reduce viral replication. They demonstrated that actually these inhibitors are targeting these kinases for inhibition of viral replication. Some of them are being already FDA approved for, again, some cancer treatments, or they are in phase 2 clinical trials for ovarian cancer. So they are non-compounds that could be tried to be used in clinical trials. And the last kinase that we found that is important is the casein kinase 2, which not only was found to be activated during viral infection, but also there is an interaction of this kinase with the M protein. And it seems to be important for the exit of the virus. And then an inhibitor of this kinase, again, is able to inhibit viral replication, so with around micromolar concentrations. All right, so in summary, what I've shown you is that the SARS-CoV-2 interactome phosphoproteome provides with rational therapeutic targets for drug repurposing. And we have identified inhibitors of sigma receptors, of translational factors, and of protein kinases as potent inhibitors of SARS-CoV-2 replication in vitro. However, we need to study this more, both clinically and preclinically. So we are now going into animal models to study some of these compounds. And we also should have in mind that it's probably better if we do combined therapies, because the use of one inhibitor, it may be difficult with one inhibitor make an impact, but if you combine two inhibitors, especially they have synergistic action, then that's probably a better antiviral therapy. Okay, so this is about our work on antivirals, and I will touch a little bit in our work on vaccines. So where are we now with COVID-19 vaccines? Well, we know that vaccines are being made in record time for vaccine development. Still, we would like to have them now. We don't have them now, but hopefully that we are very close to have vaccines that are being used, that will be used in humans. And this has been a record time in terms of generation of these vaccines. There are many candidates in the pipeline. There are more than 100 vaccines that are in the pipeline to be tested in humans. And many candidates are already in the clinic. We know about the good results now in phase 3 of the RNA vaccines from Moderna and from Pfizer. We know also good results from the AstraZeneca adenovirus-based vaccines. It looks also that the Chinese and Russian vaccines based on adenovirus are working as well. So there are very encouraging results from both preclinical models, so protection in animal models using these vaccines, and phase 1, 2, and 3 trials. They seem to have efficacy for protection against clinical disease, and they induce high levels of neutralizing antibodies in blood of people that are being vaccinated. But there are still many open questions. So first of all, we still are not sure what are the levels of antibodies in sera that are protective. So we know that they induce high levels of antibodies, but there is also a difference between some people with more or less, which one is actually the level of antibodies that is protective. Most of the vaccines are being given intramuscularly. Do we need some mucosal immunity? And the mucosal immunity in the mucosa, respiratory mucosa, in order to be to prevent infection. And we may prevent disease, but we like also, if possible, prevent infection. So, and we don't know how good they are on that when they are given intramuscularly. What is the duration of the antibody response? We hope that they're going to be long-lived antibody responses, but still we don't know that. And maybe it's needed to be revaccinated even if the vaccines are working well. Still, now for the new vaccines that are coming, now it's becoming more and more difficult to conduct phase 3 clinical trials because there are not so many places where you can do it. And then how to quickly manufacture number of doses required, and who should be the first priority groups for immunization. This is something that now is being trying to be decided. Finally, the important thing, what will be the acceptance? Hopefully there will be people, most of the people will like to be vaccinated and will be vaccinated, but perhaps there is a lot of reluctance between the public to be vaccinated with some of these vaccines, in which case we may not obtain the results that we may want with these vaccines in terms of stopping the pandemic. Well, I'm going to talk to you about our approach, which is based on a negative strand RNA virus vector, which is called Newcastle disease virus. This is an avian paramyxovirus, not a human virus. It's a bird virus. And why we think this virus could be interesting as a viral vector. First of all, a little bit about the virus. The virus produce a fatal viral disease in birds, especially in chickens. And then there is a concern in the chicken industry because they can cause a death rate of nearly 100%. And because of that, there have been live attenuated vaccines based on attenuated strains of Newcastle disease virus that are being used in the poultry industry to vaccinate chickens. So it's one of these live attenuated virus that we are using as a vaccine platform now to induce immune responses against different pathogens, including SARS-CoV-2. Now, the other interesting thing about this virus is that it's been proposed to potentially be used as for the treatment of tumors. So this is a virus that does not grow well in normal cells, human cells, but it kills human cancer cells. And it has been shown in preclinical models of cancer, especially mice, that is able to reduce cancer growth and eliminate cancers from mice that by treatment with this virus, tumors with this virus. And because of that, it has been already been used in humans as a potential anti-tumor agent. And then we know that administration in humans does not result in adverse events. So we know it's a safe virus to be used in humans. And then finally, it has some interesting properties of vaccine vectors, like it's an RNA virus, so it will not be able to integrate into host cell DNA and not causing potential problems because of that. Again, it has a good profile of safety in humans. There is no pre-existing immune history in the human population, so the virus will not be inhibited in order to induce immune response against antigens that you express in the virus. And it's very stable in terms of stability of when you insert the foreign gene. So then in collaboration with groups of Peter Palese and Florian Kramer here in Mount Sinai, we are trying to develop a Newcastle disease virus as a vaccine vector for COVID-19. Now, why we would like this one when there are several vaccines that are already most likely to work? Well, first, what happens if you need to, if there is a problem with pre-existing immunity, especially for the adeno vaccines? At least this virus has no pre-existing immunity in humans. Now, one problem with the vaccines is how to manufacture them. These are new technologies. There are not so many places where you can do the number of doses. The interesting thing is that Newcastle disease viruses are being prepared, the vaccines based on these viruses, using the same technology that is being used for preparing influenza virus vaccines. So that means that they can be easily manufactured, and there are multiple places where manufacturing can occur to obtain the number of doses that are required for vaccinating a lot of people. And then again, we know that this has a good safety profile. All right, so what we have done is, so this is the genome of the Newcastle disease virus, one of the vaccine strains, so it's an attenuated vaccine strain, and we have now inserted into the genome one new gene encoding the SARS-CoV-2 protein, a spike protein. And in general, we like to put it in such a way that we put the cytoplasmic and transmembrane domain of this spike protein, which is a membrane protein, fused to the same glycoprotein that the virus has, Newcastle disease. And this assures, when you get the virus, rescue, which is rescuing and grown in embryonic eggs, this assures that you get now incorporation of the spike of SARS-CoV-2 into the envelope. So it does not only express the SARS coronavirus 2, but now SARS coronavirus 2 spike protein will be incorporated into the envelope of the virus. And we think that that's good for being used as vaccine. So our first studies, first we compared just the full-length SARS coronavirus 2 spike versus one that contains this transmembrane domain and cytoplasmic tail based on the F protein of Newcastle disease virus, but still the antigenicity is not being changed. And we also, many of the vaccines that they are being produced also for this that are based on the spike, deleted the this multi-basic cleavage site of the spike, which makes the spike more stable, and that may be important in order to have a good antigen. So we make now these viruses. These viruses, they grow well in embryonic chicken eggs, so we can produce them, and they express nicely the spike. You can notice that if you put the wild type spike, you get the spike is cleaved into S1, and there's no antibody here against S2, and that's why we don't see it. But if you eliminate this polybasic cleavage site, it's been expressed as S0, which is more stable than this S1, S2. So the first thing that we did, and that's studies from Wina San, a postdoc in Peter Pales's lab, is an intranasal vaccination experiment in which we prime mice intranasally with this Newcastle disease virus, and then we boost it intranasally 6 weeks later. And then we prime with the different versions of this Newcastle disease virus, either expressing a spike or expressing a spike with the cytoplasmic tail and transmembrane domain of the F protein, or a different version of the virus that is a little bit more immunogenic. And we compare it also if we express only the RBD domain of the spike, which is the important one for induction of neutralizing antibodies. And then when we look at week 6, just before boosting, for induction of antibodies, one can see that all the viruses induce antibodies, except the one that contains the RBD. So the RBD is not a very good vector alone, but if they express the full-length spike, you get nice levels of antibodies, especially if you express these spikes with the chimeric, with the cytoplasmic and transmembrane domain of F, because the full-length spike, there were some mice that didn't seroconvert, 3 in this case, out of the group. So it looks like it's inducing antibodies. Then we did also intramuscular immunizations, not only intranasal. This is one in which we did an intramuscular immunization. And in this case, now we are challenging the mice with a mouse-adapted SARS-CoV-2 from Ralph Baric. And then again, we have here different groups with the different things. And then when we look to the antibodies against the spike induced in these mice after prime and after boost, there are high levels of antibodies induced by all the viruses. And if we look to neutralization, especially by the ones that they have the cytoplasmic tail of the transmembrane domain of the F of NDV, we get also nice levels of antibodies after boost day 29 post-infection with both 10 micrograms and 50 micrograms of vaccine. And now we do a challenge with SARS-CoV-2 mouse adapted. And what you can see here in non-vaccinated mice, the virus grows in the lungs of these animals, around 10 to the 4 PFU, but in all the vaccinated mice, we don't detect any virus at day 3 post-infection. And the same thing, we do a staining for antigens in the lungs of these animals that are challenged with SARS-CoV-2. You see in the control mice, you see a staining for antibodies, meaning they have the SARS-CoV-2 virus-infected cells in the lung, in both controls with anti-vector or with PBS, but the ones that are vaccinated with these different constructs, they have lungs are clear of infection. We don't see cells infected with the virus, so they are protected. Now, this is intramuscularly, going back to intranasally again, the same mice that we prime first, then we boost it now. And then in this case, we use a different system, how to infect these mice. It is for SARS-CoV-2 that is mouse adapted. We sensitize these mice by making them expressing human AC2 in the lung using an adenovirus vector, and then we challenge with wild types SARS-CoV. And when we do that, again, we see, especially in the groups that have immunized with these version of the spike that incorporated into the virus particle. We see a nice level of serum IgG at both week 6 and week 10. And then when we look to lung titers in these animals, you get day 2 or day 5, you get a nice reduction, and especially the ones that are immunized with this particular version of the vaccine, we don't detect virus replication. So it looks like it's working also in this different model, both intranasally and intramuscularly. Now, one potential concept how to use this in humans is you take the virus, you grow it in the myelinoid eggs, the same thing that you grow the influenza virus vaccine, and then you inactivate the virus. So use an inactivated vaccine that contains the spike into the envelope, and then hopefully this will be able to induce enough levels of spike antibodies in people so they will become protected. So for that, we tested this using an activated vaccine. The first ones that you see before was live. This is inactivated intramuscularly twice and then challenged against with the mouse-adapted SARS coronavirus. And in this case, we use either inactivated or inactivated with different adjuvants, because in general, inactivated vaccines, they work better with different adjuvants. These are adjuvants that are being approved for the use in humans, either DOTAP or AdavaX, which will be the equivalent for MF59. And what you see is that in all the cases, we get nice induction of antibodies, especially the use of adjuvant. What it does, it reduces the amount of vaccine that you want to put. So even with 0.2 micrograms, you get still an antibody response that is comparable to the antibody response that you get with 5 micrograms of an adjuvanted vaccine. So the use of an adjuvant allows also to produce more doses, 10 times more doses, basically, that have the same immunogenicity that without adjuvant. And then if we challenge these mice, again, what we find is that especially the ones that have using 1 microgram or 5 microgram doses with adjuvant, you see nice reduction again in viral replication. And with AdavaX, even with 0.2 micrograms, we don't detect any virus in the mouse, in the lungs of these animals that have been challenged, meaning that they are protected. And now the last construct we have used is we stabilize it even further, the spike, by insertion of what is called 6-prolines in this area. That makes the spike even more stable. And then we make now viruses that contains this hexapro spike. These are the levels of spike that we get in the virion. You can see it actually in a Comasi staining of purified NDVs. You look to NDV Lasota, you see here the L protein, the HN, the N, the P, and the M. But if you express the spike, now you see this extra band here, which is actually at nice levels, comparable to one of the glycoproteins of the virus, the HN, that is incorporated into the virus envelope. So that's good news. And then when we now do it another immunization, in this case in mice, we found that actually the one that contains the hexapro compared with the previous version, they induce higher levels of antibodies against the spike. So we think that that's probably even a better antigen. And this is the version that now we are moving to phase 1 clinical trials. So in summary, I saw you that intranasal and intramuscular immunizations of mice with live Newcastle disease virus vector and intramuscular immunizations with inactivated adjuvanted Newcastle disease vector that express the spike protein of SARS-CoV-2, induce high titers of serum IgG, and reduce viral titers in the lung in the mouse model after challenge. These vectors expressing the spike F camera induce higher spike-specific antibodies and confer better protection that if you don't have this camera, and especially the hexapro construct induce higher serum IgG than the ones that is not hexapro. And this, we have a couple of bioarchives preprints that show this data. And just to acknowledge the people in my lab, I also acknowledge all the people and collaborators, but this is a picture that we have from the lab just before the pandemic came. Funding from many multiple agencies, including NIAID, DOD, DARPA, and some philanthropy agents. And thanks to all collaborators, for people in Mount Sinai that we've been collaborating with, and also all the patients that I didn't show any of the data that we have with patient people, but we have also been conducted studies in patients to figure out what determinants are the ones that they lead to severe disease. And we're also very thankful for the patients that have contributed with samples for these studies. And again, we are in a rocket model